Thank you. Okay. Aloha, everyone. My name is Kaelyn Papako, and I'm a senior in the Honors Independent Research class. I'm excited to present my research that uses whole genome sequencing to identify genetic variation between morphotypes and environmental niches of non-tuberculous mycobacteria, or NTM, from Hawaii. Non-tuberculous mycobacteria are environmental opportunistic pathogens that can cause serious pulmonary disease in individuals with pre-existing conditions, such as cystic fibrosis. These vulnerabilities compromise mucociliary clearance, making it easier for NTM to colonize the lungs and establish persistent infection. NTM includes mycobacteria that do not cause tuberculosis or leprosy, yet they pose a growing public health threat. Although traditionally considered rare, the incidence of NTM pulmonary disease has risen significantly. Hawaii, in particular, has the highest isolation rate of NTM in the United States, with 396 cases per 100,000 persons. Despite being largely understudied, NTM pulmonary disease imposes a significant financial strain on the nation. In 2010, treatment incurred approximately $815 million in direct care costs. Mycobacterium abscessus is one of the most pathogenic NTM species, characterized by its ability to evade host immune defenses and its antibiotic resistance. Infections caused by M. abscessus are often associated with aggressive disease progression, yet limited therapeutic options are available. As such, understanding the unique biological mechanisms that drive M. abscessus, such as its morphotype switching, is essential to developing more effective interventions and improving patient outcomes. A unique characteristic that has been recognized in some species of NTM, including M. abscessus, is the ability to display two different colony morphotypes, smooth and rough. It has been suggested that the mechanisms of infection are what drive the transition between smooth and rough through the loss of cell wall lipids, namely glycopeptidolipids or GPLs in its cell wall. The smooth morphotype contains high levels of GPL in its cell wall. On the other hand, the rough morphotype has reduced GPLs and displays cording, which is a characteristic associated with increased virulence. The loss of GPLs exposes surface molecules that trigger a stronger immune response, therefore contributing to more aggressive clinical manifestations. Rough morphology of M. abscessus is significantly associated with immunosuppression, cavitary pulmonary disease, and poor clinical outcomes. Whole genome sequencing can be used to identify genes associated with GPL biosynthesis in NTM. Among the notable findings from whole genome sequencing studies is a large cluster of 19 genes in M. abscessus that plays a central role in GPL production. Several genes within this cluster carry out specific biochemical transformations crucial to the structural integrity and function of GPLs. Despite rising infection rates and the lack of effective treatments, NTM remains understudied, especially in Hawaii. Scientists still don't fully understand what causes NTM to exhibit rough or smooth morphology, though it's believed to be linked to GPL biosynthesis. I hypothesize that whole genome sequencing could help identify genetic differences between smooth and rough NTM, providing a deeper genomic understanding of NTM morphology. In collaboration with the Honda Laboratory at the University of Texas at Tyler, we created a panel of 8 environmental M. abscessus isolates. Included in this panel are 4 pairs of smooth and rough isolates from soil and wells on Hawaii Island and Oahu. Soil samples collected from the same location share the same number. For example, soil 1S and soil 1R originate from the same site, with one exhibiting a smooth morphotype and the other a rough morphotype. After collection, DNA was extracted and sequenced using the Oxford Nanopore Technologies MinION platform. Reads were assembled using Fly, and genome completeness was assessed using CheckM. The assembled genomes were then annotated with BACTA. Then I extracted GPL biosynthesis gene sequences from GenBank and aligned them to our NTM genomes in Genius. To further analyze genetic diversity, I conducted a pangenome analysis. The initial pangenome analysis revealed that gene presence and absence clustered more strongly by environmental origin rather than by colony morphology. The presence absence matrix indicates clear genetic distinctions based on environmental source. However, no consistent gene clusters could be exclusively linked to either rough or smooth morphotypes. The well isolates formed its own clade, while the soil isolates also formed its own clade. This suggests that genomic content does not directly drive morphological differences and instead may reflect adaptation to environmental niches. The implication is that the phenotypic switch may be governed by a smaller subset of genes or specific mutations rather than large-scale differences in overall gene content. The pangenome analysis also accounts for gene presence and absence across the cloud, shell, and persistent genomes. The cloud genome includes genes unique to one or two strains. The shell genome contains genes shared by two or more strains, and the persistent genome comprises genes found in all strains. All samples, regardless of their environmental origin, share the majority of genes within the persistent genome. However, a few genes are exclusive to either well samples or soil samples, potentially reflecting the distinctive selective pressures of their respective environments. Based on the amino acid alignment, several mutations in the RMLA gene were observed in specific isolates clustering according to morphology. The RMLA gene was selected after comparing the sequences of 19 genes, as it showed the most distinct differences between rough and smooth morphotypes. The green indicates 100% identity. Green-brown regions indicate greater than 30 but less than 100% identity, and red indicates less than 30% identity. Although most of the RMLA sequence remains highly conserved, there were a few key residue differences between smooth and rough isolates. Well 1S, 1R, soil 2R, and soil 3R share nearly identical gene sequences, while soil 1S and 1R, soil 2S, and soil 3S all exhibit highly similar gene sequences. For well 1 and soil 1 pairs, the similarity in gene sequences is likely due to their shared environmental conditions rather than differences in morphology. Our MLA encodes D-glucose 1-phosphate thymidyltransferase, which catalyzes the first step in L-rhamnose synthesis, linking the glycolic acid layer to the peptoglycan core of the mycobacterial cell wall. Disruptions in RMLA function may impair L-rhamnose and GPL production, affecting the cell envelope's integrity and immunogenicity, which is a key factor in M. abscessus, where rough variants with reduced GPL show higher virulence and worse clinical outcomes. Amino acid substitutions may alter RMLA function by changing charge or hydrophobicity at key sites, which impairs substrate binding or product release and promotes a shift towards a rough phenotype by limiting GPL formation. Mutations in our dataset may thus serve as molecular switches modulating enzyme activity. Through my research, I found that pangenome analysis shows smooth-rough pairs cluster by environmental origin rather than morphotype, suggesting that key differences may reside in specific genes. This discovery led us to the RMLA gene, where we identified distinct mutations and indels associated with each morphotype. By identifying a key mutation in the RMLA gene that may drive the smooth to rough morphotype switch, this research advances our understanding of the genetic mechanisms underlying NTM pulmonary disease and opens new avenues for developing targeted diagnostic and therapeutic strategies. Limitations include a small sample size of 8 isolates and potential sequencing errors and biases. Future research should involve structural modeling to determine how these mutations impact protein function, and gene expression studies to assess whether RMLA is differentially expressed in smooth versus rough NTM. Through independent research, I developed essential research skills like how to conduct a literature review, how to develop a research plan, and how to perform advanced lab techniques. This project also has guided my future career goals, igniting an interest in the intersection between microbiology and human health, with an emphasis on Hawaii-based research. So, to future research students, choose a topic you are interested in and do not doubt your abilities. And I'd like to extend a huge mahalo to my mentors, Dr. Jennifer Honda, Miss Rachel Wilsey, Dr. Ling Hao Ding, and the Honda Lab for guiding me through my project. Also to Dr. Chan, Mrs. Kobayashi, and Mr. Hill for aiding me with my research plan, lab work, and data analysis. And finally, thank you to my family and friends for supporting me through this process. Mahalo.